drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Studies?
Sure. So I'm a professor in the Department of Microbiology and Immunology at the University of California in San Francisco. And our lab is kind of broadly interested in how the gut microbiome, the trillions of microbes that are found within our GI tract, influence both nutrition and pharmacology.
Yeah, and so you study lots of interesting stuff. There's many different directions we could take this conversation. I've covered the microbiome a lot on this podcast. I've covered diet stuff a lot on this podcast. I want to start off pretty high level and get you talking about something that doesn't necessarily have to do with your work specifically, but it's sort of broader questions related to diet and nutrition and how it's studied in the literature. And it obviously ties into the work you do, but I want to talk about some of the different types of diets that are out there that are commonly studied, and some confusion and shortcomings maybe even the literature has in terms of how those things are studied and how they're talked about. So the place I want to start is I'm going to keep it vague intentionally. One of the most common diets that you'll read if you dig into the scientific literature that has to do with diet is the so-called high-fat diet. And maybe some of the listeners will know from listening to me before, but this is one of my biggest frustrations in the literature, this term high fat diet. What can you tell us about the high fat diet and what that term maybe doesn't tell us, or problems that come from labeling and using diets with that level of specificity?
Yeah, that's a great question and kind of near and dear to my heart. I don't know if you saw, but we had a paper a few years ago where we did a meta-analysis where we were interested in kind of comparing the microbiome profiles, mostly in mice, but also a little bit in humans, across the hundreds of studies where people have fed mice some kind of form of a high-fat diet compared to a control diet. And so it was something we thought about a lot in that study. But to your question, I guess going back to my work in grad school, we're kind of careful to use the term high fat, high sugar diet because we thought, especially when you think about the microbiome, it's important to keep in mind the kind of other macronutrient components. And so typically when people model obesity in mice, they give them a diet that's not only high in fat, but also really enriched for sucrose and maltodextrin, relatively simple carbohydrates that are readily digested by the host.
But
and
importantly,
then you're kind of stripping out a lot of the fiber or complex carbohydrate.
But importantly, when they give them these diets that are high in fat and high in these other carbohydrates, they simply usually call it high fat diet, right?
Right, exactly. Yeah.
Okay.
Yeah, typically like HFD or like high fat diet is the acronym, which is misleading, for the point you make, which is that it's actually different in terms of the types of carbohydrates. But even kind of more profoundly, something that I think a lot of people don't realize is that, normally when we study lab mice, they're effectively eating whole foods, given that it's largely soy and corn and other kind of commodity crops that are used to kind of smash together into these pellets, as opposed to your typical high-fat diet, which is actually made from purified ingredients. So it's more like a processed food that you might buy in the grocery store. And so, we think in terms of how the microbiome responds to these diet shifts, it's really important whether or not they're kind of seeing the nutrient in the context of a real food as opposed to just a purified sugar.
Yeah. So when we read high fat diet in the literature, more often than not, it's not merely a high fat diet. It's high fat plus high carbohydrate. And if you compare, there's hundreds, if not thousands, probably thousands of studies that use the so-called high fat diet, or at least they use that nomenclature, but they can
Hmm.
vary tremendously, not just in whether or not they also have high carbohydrate or high sugar content, but also the specific types of fats they use
Hmm.
and all sorts of different things. And so it's a bit confusing because, if you were to just look at the literature and say, let's just look at all the studies that use a quote high fat diet and see what they say, it's actually not, it's just not appropriate to sort of like look at the one vector of where all those studies point, because it's really many different diets that are simply, we're using the same words to describe.
Yeah, totally. And that gets really kind of complex, especially if like every tiny variation matters for what
Mm-hmm.
you're studying. In our case, the microbiome, but others might be interested in, body weight or other host phenotypes. And I think one way that we've graphed this is to you can kind of have a triangle where, one corner is a diet with 100% fat, and then the other corner is 100% protein, and then the third is 100% carb. And then that allows you to kind of, in that um, triangular space, kind of plot where each diet falls. And
Yeah.
in the meta-analysis that we did to look at the gut microbiome, what was really striking is kind of how variable the high fat diets were in terms of not only like how far they pushed the fat intake, but also whether or not protein and carb changed.
Yeah.
And, that's at the simplest level when all you care about is level of macronutrients and we're not worrying about the type of fat or type of carbohydrate. Um, and then you can even find examples where the control diet is kind of higher in fat um, than some of the high fat diets.
Wow. Yeah.
And so, it means that, it's like really important, to take into account exactly what you're comparing when you want to, see whether or not you see the same effects study to study.
Yeah.
Um,
Yeah. So you're saying there's studies where, like, study A might use a high fat diet and study B has a control diet, what they call a non high fat diet, and it might actually have
right.
higher fat content than the other study that says high fat diet.
Yeah, like my low fat diet might be higher in fat than your high fat diet.
Right, right, right.
Because like typically I mean, it makes sense how we get here because people are using these terms in the context of two diets that are in their study, right?
Yeah, yeah,
So
yeah,
like really they're just trying to say that one is higher and one is lower.
yeah,
But it's kind of tempting to apply that between studies.
yeah, yeah.
Um,
And then like one thing that's obviously becoming more popular, and getting more attention in the research world is the ketogenic diet. So if we get a little bit more
mhm,
specific with how we describe these things, high fat diets may or may not be ketogenic. So a ketogenic diet is a high fat diet, but when we read high fat diet in the literature, broadly speaking, those are often not ketogenic, whether or not they have high fat, whatever that means exactly. And they also often have high carb diets. Can you just talk a little bit so is it fair to say that the majority of the time when a study says high fat diet, it's probably pretty high in both fat and carbs and therefore not ketogenic?
Yeah, yeah, definitely. And you're kind of like reliving the journey that I went through, I guess, starting in grad school and then continuing today in the lab. And I guess just to kind of finish the thought on the meta-analysis, that I think one of the kind of really what I thought was good news pieces of data that came out of this is that, even though there is this tremendous variation in the types of low fat and high fat diets that are used in the literature, when we were able to kind of combine microbiome profiles from all of these different studies, um, we were still able to capture really reproducible effects on the gut microbiota. And so, that suggests that really, the microbiota is responding to a large extent, to these general shifts in macronutrient intake, and that they don't, even though, maybe at a quantitative level they can sense the type of fat. Um, but that the kind of general trends are able to transcend the specific flavor of high fat diet. And so I think that was good news, and kind of like, it suggests that you might be able to translate some of this to humans, where there is no kind of single high fat diet in the human population. We're all eating variable diets
Hmm.
to each
Hmm.
other. And so hopefully, we can kind of find these, more foundational ways in which the microbiome responds to a shift in macronutrient intake.
Yeah, and
Um,
if we think in terms of this sort of triangle you described of macronutrients, so you've
mhm,
got one corner is carbohydrates, one's fat, one's protein. A given diet's going to fall somewhere in that space. It might be high in
mhm.
one, low in the other two, or vice versa, or high in two and low in one, whatever. Can you give us a sense, a very, very basic sense for like when we talk about the standard American diet, the diet that's going to be fairly representative of what the average person in the US is eating, where would it fall in that triangle? Is it high carb and high fat, low protein, or where exactly would we place it?
Yeah, and yeah, I think it varies study to study, but it's somewhere in the realm of like 45%, um, kilocalories from fat in the kind of standard American diet. And that was one of the things that we, I guess, continue to think a lot about with respect to mouse models, because that I think one of the challenges in translating from mouse to human is that if you take your average American, they're already on, at least in terms of macronutrients, the equivalent of a mouse high fat diet.
Mm-hmm.
Um,
So that would
and
be a
so,
non-ketogenic diet with high fat levels, but also probably high
right.
carbohydrate levels. Yeah, yeah,
And because mice normally on the standard mouse chow consume such tiny amounts of fat, um, if you want to kind of induce the same level of shift in macronutrients, you have to go in humans from a high fat to a ketogenic diet.
yeah.
And so it kind of, matters depending on like what the baseline intake of the population is.
Mm-hmm. Mm-hmm.
Um, and so, I think in Americans, it's like, we can do studies readily to either, to lower fat intake, but it's difficult kind of with normal diets to go higher than what a lot of people are already eating.
Mm-hmm. Mm-hmm. And what would be just to spell it out for people that aren't familiar, if we take a high fat, meaning high fat, high carb diet, and we compare it to a ketogenic diet, what are the differences there, both in the composition of the diet and in terms of the kinds of metabolic shifts that would induce in an animal?
Yeah. So, the big kind of famous shift that happens in terms of a ketogenic diet is that you really, have to lower carbohydrate intake dramatically. And so, when the body kind of shifts from using glucose as its primary fuel to lipids or fat, um, that allows the release of the what are called ketone bodies that go into general circulation and then provide fuel for different tissues. And so that requires kind of the combination of very typically very high levels of fat and then very low carbohydrate intake. In humans it varies, and then in mice we typically remove all of the carbohydrates, and so they're really only eating fat and protein.
Do is there anything? This is a topic that I don't hear covered a lot in the literature. So obviously, mice are a convenient model for all the
Mhm.
reasons that people use mice. When we think about them in sort of evolutionary and ecological context, are there wild mice that eat truly high fat or ketogenic diets? Or I guess another way of asking what I'm asking is, would we expect mice to have the same degree of kind of metabolic flexibility that a human being has in terms of its ability to adapt to very large shifts in the fat and carbohydrate content of its diet?
Yeah, it's a great question. It's not something we really looked at in terms of kind of high fat consuming mice, but um, yeah, I mean, we did have one study where we kind of tracked wild mice throughout the course of a year, and this was working with Amy Peterson and her colleagues in the UK, where in the UK they have these beautiful study sites where it's kind of wild, fields where they've gridded off different areas to capture and release mice throughout the course of a year, and this has gone on for decades.
Oh, wow.
And so you have the ability to kind of like follow the same mouse over time as they switch from eating, different foods depending on the type of the time of the year. And kind of as expected, we did see a pretty clear shift in the microbiota of these wild mice that we could kind of correlate to the change in, eating seeds versus insects primarily. Um, but it's messy, right? Because they,
Yeah.
just like in humans, they still have access to different options, and so we don't see the kind of like clear transitions that you might see in a laboratory mouse.
Yeah. Yeah.
Um,
So they will adjust their
and then
diet
and
seasonally.
then yeah, yeah, and I think that is kind of s- since then people have reported similar kind of seasonal effects in the human microbiome, and so I think that's probably a general kind of feature of animals that are experiencing different nutrients at different times of the year.
Yeah. Yeah.
Um, but the other thing that we're really interested in now is like thinking about more exotic diets that wild animals consume. And so, for example, we've done a lot of work on this drug digoxin, which is a natural product isolated from the foxglove plant. And foxgloves are really toxic to most mammals, but
Hmm.
interestingly, mice are resistant to them. And so we're doing some studies with Denise Deering and other collaborators to kind of track what happens to the microbes in the gut of mice that are kind of foraging on these really toxic plants that humans can't consume.
Hmm. But earlier you said when we give a ketogenic diet, a diet that can induce ketosis in a mouse, you typically have to pretty much just remove all of the carbohydrate. Is does that mean that it's basically harder to get a mouse into ketosis than a human?
Yeah, I definitely think that's true. So yeah, not only is kind of starting the ketogenic process or ketogenesis more challenging, but also mice. Yeah, I mean this is true for a lot of things in mice that they're very lab mice. Even though you might expect them to be wimpy, are often really resistant to a lot of the diseases that we have, and so. Um, we think this is definitely true for metabolic diseases like diabetes and obesity,
Hmm.
as well as other diseases like autoimmune disease. You kind of have to this is why we have to leverage
You
kind
have
of
to really
special
push
mouse
them.
models. Yeah.
Is that I I'm just guessing, so you tell me if I'm right or wrong, but I'm guessing that sort of acts, is that just a byproduct of breeding mice to, to live and survive in the lab, and as a side effect, they're actually hardy in certain ways?
I don't know. Yeah, it's a good question. I mean, maybe one of the reasons is like we tend to study younger mice in the
Mm.
lab. Um, no one, although there's exceptions, but it's hard to study a middle-aged or an older mouse.
Mhm, mhm.
Um, and so, some of these diseases do kind of progressively get worse if you look in an older lab mouse. Um, but yeah, I think it's an interesting question. I don't know, if you look in wild mouse populations, whether or not you can find more evidence of disease. I
Mhm.
think like one of the things that was really clear in the study that we did with Amy is that um, mice in the wild are really, they have a very hard life. And so, I think before any of these chronic diseases happen, they're getting infections and they're getting killed by predators.
Right,
And
right.
then over the winter, at least in the mice we were studying, there's a huge population crash. And
Yeah, they
so,
starve.
yeah. So, I think there's a lot of other forces that kind of like are acting against even being able to look for these other
Yeah,
diseases. But,
yeah. But like, one of the things I wanted people to take away from this part of the discussion is obviously mice aren't humans, but there's all sorts of other things that were, there's issues with the literature in terms of how we speak about things, how we set up the experiments that
mhm.
start to lead people astray in the sense that when you'll often see, even with scientists, sweeping claims about the high fat diet does this, and they're inferring
Right.
things from rodent studies where all the high fat diets are different from each other, and then mapping that onto humans. And there's so many like little jumps and assumptions that get made along the way that people often don't realize are being made. One of the things I liked about some of the work that you've done is you've started to fill in some of the blanks here. So one of the studies I wanted to talk to you about was, people are interested in high fat, low fat. People are interested in animal based, plant based, high fiber, low fiber, all these different types of questions, how it affects the microbiome and so forth. You've actually done, well, I'm going to ask you about one study. There's more things I'm sure we can talk about, and I'll let you sort of steer us there. But
Mhm.
you had this 2014 paper that I thought was really interesting. You actually took human beings and you gave one a plant based diet that had high carb levels, so that's not going to be ketogenic. One had an animal based diet that did put people into ketosis, and you actually measured some things across these
Mhm.
people. Can you start to talk about the setup for that study and what the motivation was for it?
Sure. Yeah, I think a colleague of mine called it carnival diets or something. I don't know if it was in derogatory, but yeah, I mean, I think we've kind of been alluding to the logic that got us there, and so we I when I was in grad school, like one of the what I thought was a really exciting finding was that we took lab mice and we put them on a high fat, high sugar diet, um, and compared the microbes in their gut to low fat controls. And then I don't know if you how quickly, or you probably know the answer now, but how quickly you think the microbiome would change if you go on a new diet. But
Well, yeah, I'm spoiled because I know the answer, but so this to set you up for this. One question there was like, obviously your microbiome is going to change to your diet, but a big question was how much and how quickly.
Right, yeah,
Yeah.
yeah. And
And so,
that was the really striking thing in these initial mouse experiments is that, I kind of imagined it would be slower and, we
yeah,
might see more gradual effects that
takes weeks to
compound.
adapt to it. It's sort of the slow
Yeah,
roll, maybe.
right. But, instead what we saw was just after a single day, it's kind of like a light switch. You can see you go from one configuration of the gut microbiota to another.
Wow. So
And so
literally,
it's this.
literally in one day.
Yeah. And so, it was a little bit of a bummer because I spent a lot of time collecting all these other time points. And so that, I mean, it got a little stronger throughout week one, but then, like really all the action was in the first week, at least in the mouse model. And so, uh, that kind of raised the question of whether or not this could happen in humans. Are our microbes actually changing every day as we decide what to eat? Um, and it kind of conflicts with some of the more traditional views of the gut microbiome is that it's like very stable throughout adulthood and that,
Yeah.
there's not a lot of changes over
Yeah.
time.
And I guess
Um,
one thing to unpack there is that statement isn't coming out of nowhere. I think when you measure the microbiomes of a typical person, it will seem pretty stable day to day, week to week, but that's probably an artifact of their lifestyle and their diet being relatively stable.
yeah, yeah, well, I think multiple things that contribute to that. So one is that like, and then this kind of becomes semantics, but it's really important how you define stability.
Mm-hmm.
And so, like one of the breakthroughs, kind of, um, early on in the microbiome field were the development of all these beta diversity, like ways of comparing microbial communities to each other. And so that allows you to like plot microbial communities on a PCOA space or some other dimensionality reduction method. And, but typically what you're comparing is multiple people.
Mm-hmm.
So, you have samples of one person over time and then a second person. And if you plot them on a PCOA, typically the samples from the two people stay apart from each other so that you kind of retain that, the signal of individuality.
Yeah. So they're
Um,
they're different from each other, but that doesn't necessarily mean each one is static within the person.
right, exactly.
Yeah.
Yeah, like within each person, there can be dramatic fluctuations, but as long as my microbiota doesn't become yours,
Yeah.
we're still going to end up apart on the plot. And so,
Mm-hmm.
um, yeah, it kind of depends on what the measurement is. And then, yeah, and then to your other point, I think, Yeah, I guess I hadn't appreciated when we got into this stuff that like, although maybe I should have because my diet is pretty boring. But like people really don't change what they eat that much, I mean, we do go to restaurants, so that's a source of chaos, but
Yeah, but
a
it's not
lot
like
lot of people have very routine diets.
yeah, it's not like I mean it's certainly not going to be as drastic as what you did in the study. It's not like on Monday I'm doing a vegan Monday, Tuesday, and then a carnivore
Yeah,
Wednesday or a ketogenic Friday. Like it's going to be relatively stable in terms of the overall macronutrient profile and whether I'm generally more plant-based or animal-based for most
right.
people.
Yeah, and like that turns out to be true not just for people that are on explicit diets to lose
Yeah.
weight or for some other reason, but just because, like we're habitual creatures.
Yeah. I
We
like the
tend to
foods I
have very
like.
similar breakfast and lunch.
Yeah. So anyways, like going back
Um,
to this 2014 study, what were the two diets that you used and why did you engineer them the way that you did?
yeah, so kind of the thinking was like we wanted to really push the needle and, try to change macronutrients in a big way. And so that kind of got us to this idea of like, if most of the people coming into the study are already eating a decent amount of fat, we want to get them even higher. We
Mm-hmm.
weren't really intending to study ketogenesis.
Mm.
It was just kind of a happy accident of designing a diet that would be really rich in fat. And then and then I guess like what we realized is that really the only way, one of the which is true in the kind of um weight loss diet literature, if you try to the easiest way to get to high fat is to drop carb a lot.
Mm-hmm.
Um, and so we thought, why don't we just go all the way and make it only meat,
Mm-hmm. So it's basically
So
just meat and cheese. It was an animal-based
yeah,
ketogenic diet.
right,
Meat,
yeah.
cheese,
So,
eggs.
much more extreme than what your average person on a ketogenic diet eats, but
Yeah. So it
um,
was high fat, very, very low carb, low fiber, and it's all animal-based foods.
yeah, yeah.
And then the other group was plant-based, and it was basically high carb, high fiber, a mix
Right.
of plant-based foods, but it certainly wasn't ketogenic and it certainly wasn't only fat.
Right, exactly. Yeah. Yeah, then, once we kind of like realized this would be fun to test an animal-based diet, we thought, we should do the opposite.
Yeah.
And so, the downside is like these two diets are so different from each other in so many different ways that, it's not a great design in terms of getting at mechanism, right? So
Right. You
if you
can't,
see changes, what is driving it is really hard to piece apart.
yeah, you can't conclude from the study, oh, the fiber did this, the protein content did that, the
Right.
fat content did this, but it is naturalistic in the sense that you were giving people whole foods, you were giving them diets that some people intentionally, try and eat. There's people out there trying to eat animal-based ketogenic diets.
Right.
There's people
Yep.
out there trying to maximize fiber content from plant-based whole foods and so forth. And you saw pretty big differences in terms of
Mhm.
what the response was in each group. Can you give us a basic survey of what happened to people eating the plant versus the animal based diet?
Yeah, so I think the most exciting thing for us kind of gets back to what we were talking about in these early high fat mouse experiments is that we gave people food coloring in along with their special diets for 5 days. Yeah, the design was that we had daily sampling from people for a few days before each diet and then after each diet, and then the diet each diet arm was given for 5 days. And so along with that, we gave food coloring, and that allowed us to kind of track when the first sample of the new diet came out of the gut. And using that, we could kind of estimate that actually we could see differences, especially on the animal-based diet, in kind of overall structure of the microbiome,
Mm-hmm.
just within a single day
Wow.
of being on the diet.
So
And then those, yeah,
you give
go
people
ahead.
each of these. So half the people get a plant based diet for 5 days, half get an animal based diet for 5 days. You've also got 5 days of measurement before that shift, and then after that
Mhm.
shift when they're going back and forth to their normal diet. And you're saying that in that 5 days, you see drastic changes in the microbiome, and you can actually start to pick them up within just one day.
Right, yeah, exactly. Yeah, and really like the strongest effect was going from kind of, their standard um starting diet onto the animal-based diet.
Hmm.
Although we also did, see some differences in response to the plant-based diet.
Yeah, so you see
Um,
differences in both groups. Obviously, there are going to be different differences in the plant
right.
versus the animal group, but overall, sort of the magnitude of the shift was probably stronger for the animal-based diet.
Yeah. Yeah, and I think one thing we kind of speculated that I still think is true is that um like oftentimes the study design, if you think about fiber, for example, we take people that are on a standard American diet and then try to ramp up fiber intake.
Yeah, here it's the
It's
opposite.
much more, yeah, it's much more rare that we do the opposite.
Yeah, yeah, yeah.
And so this is a diet where you're really depleting, dramatically depleting the carbohydrates that a lot of bacteria in the gut are
Yeah.
going after.
Yeah. So the people on a plant-based diet are increasing their fiber content. So they start at some baseline, and it's
Hmm.
certainly not zero. Maybe it's, some people might argue it was higher, it was low, but it was some fiber content
Hmm.
as most people would eat at baseline. That goes up in the plant-based group. And then the animal-based group, they're really getting no fiber. There's essentially
Right.
zero
Yeah.
dietary fiber. And
Hmm.
what would, before you looked at your results, what would the prediction there be on how increasing fiber versus taking it down to zero would affect people?
Yeah. Yeah. So yeah. The other thing I wanted to mention is that so that one of the really, actually the first study I worked on when I was in the Gordon lab as a graduate student, I was a brand new rotation student, assigned to work with Ruth Lay, who had come from Norm Pace's lab and was experienced with a lot of these tools for microbial ecology. Um, and Ruth, before I got there, had gone to the San Francisco Zoo and collected a bunch of poop from different animals.
Hmm.
And so the idea was, now that we had the ability to sequence microbes in the gut, we could kind of compare which microbes were in a zebra versus a giraffe versus a lion,
I see. So you could get a
versus a
sense for
human.
what a carnivore microbiome looks like versus a herbivore microbiome.
Exactly.
Yeah.
Yeah. And so, kind of one of the takeaways from that study is that you could see these correlations between, as you might expect, the carnivores and the omnivores and herbivores all grouped separately. Humans kind of looked like we were regular omnivores. At the time, it was just a handful of people, but you could kind of like group animals by diet just based on microbes in their gut.
Yeah.
And
Which
then subsequent,
makes
yeah,
intuitive sense, right?
hmm
Yeah.
and then after that, Brian Meggie, another graduate student in the lab, did metagenomic sequencing of those same samples to kind of compare the levels of different genes in the gut of carnivores and herbivores. And kind of as you might predict, the microbes are really flexible in what they can metabolize, and so when in the gut of your typical herbivore, you see a lot of genes that are involved in breaking down plant polysaccharides, so these complex carbs that are in plant products.
Yeah.
And then when you look in the carnivores, there is an enrichment for genes that are involved in breaking down proteins and amino acids and using those for kind of alternative types of bacterial fermentation. And so there is kind of, a nice kind of genetic signature of, what types of pathways might be useful to a microbe in a
Yeah.
carnivore versus an herbivore. And so that was kind of what we were thinking leading into the human study, and in many ways kind of played out, especially when we started to looking at the expression of genes in the community.
Mhm.
We could
So
see some of these kind of signatures of switching from plant fermentation to protein fermentation.
yeah. So basically you had a sense from prior work. You guys literally went to the zoo. You literally looked at
Hmm.
the gut microbiome of herbivores like zebras, carnivores, like other animals. You collected the waste from animals at the zoo, and you had a notion that certain animals on certain diets had certain microbiomes. This involved microbes with metabolism that made sense, right? If it's a
Hmm.
plant eater, it's got metabolic genes turned on and microbes present that break down plant sugars and plant fiber and things like that. And then if it's a carnivore microbiome, you've got stuff that's involved in
Hmm.
breaking down the amino acids from proteins and stuff like that. So you had a
Hmm.
sense for what like the carnivore to herbivore spectrum of the microbiome looked like.
Right.
And when you gave people these diets, broadly speaking, they conform to that directionally speaking.
Right, exactly. Yeah. And I think it's, the more you think about it, it's kind of wild that you can become a carnivore.
Yeah. Well, I mean,
That
it is kind of wild, but at the same time, I've learned a lot about this the last year or so. And to mention just one podcast that was really interesting, I had these two guys on. They're basically paleoanthropologists by training. And long story short, they studied ancient DNA and ancient tissue samples
Mhm.
from the Clovis peoples of North America during the last ice age. And they basically showed that these people were eating the same diet that saber-toothed cats were eating, which was probably a
Mm,
megafauna meat-rich ketogenic diet.
interesting.
So people in
Yeah.
history, some of them have eaten that type of diet.
Right, yeah. Yeah, and I think what our data suggests is that your microbes are really plastic and that
Yeah.
they can adapt to these changes rapidly. And so, it raises a lot of interesting kind of evolutionary questions that we're not really working so much on in our lab, but Rachel Carmody, a former postdoc from our lab, thinks a lot about
Yeah. When you look
um,
at, I mean, when you,
the potential for microbes to kind of open up new access to nutrients as they adapt to what
yeah,
humans are trying.
yeah, I mean, look, one of the things your study clearly shows is, as you said, you see big changes in the gut microbiome composition to both types of diets in different directions in 5 days. You start to see it within one day. And that fits with the idea that humans are just very metabolically flexible. And when you start to think
Hmm.
about it like holistically, when you just look at the variety of diets out there today, the variety of cultures
Hmm.
and cuisines out there today, and the stuff I mentioned before and other stuff from the paleo literature, humans have eaten all sorts of different diets, and that's enabled us to go into, basically every corner of the globe. So it makes perfect sense that a feature of our species would be metabolic flexibility, and that would
Right.
imply gut microbiome adaptability as well.
Hmm. Yeah, and I think that the kind of diversity of the gut microbiota allows, and not only kind of this like flexibility where microbes that are really well adapted to different types of diets can bloom or decrease in abundance. But also, nice work from Justin Sonnenberg and others have shown that even within a single species, microbes, the bacteria can kind of turn on and off different pathways, and so
Yeah, yeah,
they have the ability to kind of change what they're eating depending on what we eat.
yeah. So it's not just that our microbiome can shift in terms of which species are present. Even you could have the same microbiome and the
Right,
same exact composition. It could switch which genes are turned on and off, and therefore its metabolic properties.
right, exactly.
One of the things I wanted to ask you about in this study. So to recap, plant-based diet, high fiber; animal-based ketogenic diet, basically no fiber. One of the most common things I see in the literature about fiber, and I want to ask you about this because I've been thinking about it. Basically, if I'm going to caricature what I see in the literature with respect to dietary
Mhm.
fiber, it's dietary fiber is good because it supports good gut health, because we have gut microbes that can digest what is otherwise undigestible plant fiber for us, and they digest it into short chain fatty acids, and that's good for our gut epithelial cells. Baked into that statement, and we can critique it in a moment, but baked into that
Mhm.
statement is the only way to get these short chain fatty acids like butyrate is from bacterial fermentation of that dietary fiber. How did this short chain fatty acid profile from the animal and the plant based diets compare in the study that you did?
Yeah, so we definitely saw differences that are consistent with that general thinking. So there's certain short chain fatty acid products like isobutyrate and isovalerate that are thought to mainly come from protein fermentation.
Mm-hmm.
And so, yeah, I guess like one thing for those that aren't like deep into this is that there's actually many short chain fatty acids,
Yeah.
and they can come from many different sources, including there's bacteria that can take hydrogen and CO2 and produce a short chain fatty acid. And so you don't even need to come from a macronutrient.
Hmm.
You can come from waste, basically, like gas waste.
Wow.
And so, it's a lot more complicated than like this kind of simple model that people typically mention, which is obviously true that fiber gets fermented and then that gives you butyrate.
Mm-hmm.
Um, but butyrate is kind of one of many different compounds that's produced.
Yeah. So there's
Um,
multiple short chain fatty acids, not just butyrate. But the other thing that I thought was interesting here is So the thing that was expected is you would expect the plant-based high fiber diet to give you more butyrate, and indeed that diet had higher butyrate levels, those
Mhm.
people did, than the ones on the ketogenic animal diet. But what I thought was interesting is the butyrate levels for people on the animal-based diet did not go to zero.
Mhm. Mhm.
Was that surprising to you?
Yeah, definitely. Yeah, and I guess it's trying to think of a specific example offhand, but yeah, not only in the human data, but like also like um I'm always surprised when you see data from, for like germ-free mice or diets that are really low in fiber that you can still detect butyrate. And so yeah, it definitely raises the question of like what the sources are. As I mentioned, like you can get acetate, you know, if you just have hydrogen and CO2, you can create acetate
Mm-hmm.
with bacteria. And then from there, there's microbes that actually interconvert acetate to butyrate.
Mm-hmm.
And so, there are kind of like fiber-free ways to get to butyrate.
Yeah, yeah,
But yeah, I think it's an open question, kind of like where these what actually the source of these compounds is
yeah,
normally.
yeah. But I
Um,
mean, the other thing that I thought was a good takeaway here is um, a lot of times in the discussions I read about or hear with respect to dietary fiber, it's the simplistic story of fiber goes to butyrate via bacterial fermentation, goes to good health outcomes. But really,
mhm.
when you shift a diet in the ways that you did, going to either a plant diet or to an animal based ketogenic diet with no fiber, you see the whole profile of short chain fatty acids in both groups. It's just the mix of them is different, but they're all there in both groups.
Yeah, yeah, and I think what part of what's happened over the last like decade or so is that I guess when I got into this, it was very much, or at least we were thinking along the lines of energetics. So it's butyrate fuels the enterocyte. Another, probably the most abundant short chain fatty acid typically is acetate.
Mm-hmm.
And acetate is interesting in that we know it can get absorbed out of the gut and then incorporated into adipose tissue. And then later papers have looked at like acetate trafficking to the brain and all sorts of other tissues. Um, so but primarily we were thinking about these as fuel, But as you may know, we now know that the short chain fatty acids can also be signaling compounds, and so
Yeah,
butyrate, for example, is an HDAC inhibitor. They also have effects on immune cells, and
yeah.
something Wendy Garrett and others discovered is the T regulatory cells that kind of provide a check on inflammation are induced by short chain fatty acids. And so, it's not as simple as just the like model of, providing more calories to gut cells.
Yeah, these things
Um,
aren't merely fuel to be used for fuel.
right.
Yeah.
hmm And then, that's not, something that's really been ignored is like what they do to the microbes,
Yeah.
it's, we, because presumably like if you drive fermentation towards butyrate, butyrate, could potentially affect other bacteria in the community, and so you can have this kind of ripple effect.
Yeah, yeah. So either a plant-based diet high in fiber or a ketogenic animal-based diet can rapidly reconfigure the gut microbiome. You see shifts in the short-chain fatty acid profile in each diet. And when you take them off the diet, do you also see like a rapid reversion right back to where they were before, or are there sort of lingering effects that prevent it from going back to what it was?
Yeah, we did. For the most part, things went back to as they were, and that was really a big relief. But yeah, it's definitely something I worry about more generally in human studies. That's not always the case. For example, in studies of antibiotics, David Rullman's lab has really nicely shown that you can have really persistent effects of antibiotic
Hmm.
exposure.
Yeah, yeah,
That can go out weeks to months after the initial after taking the drug.
yeah.
And so, you always worry that, we don't want to do human studies if we're kind of permanently changing your microbiome.
Yeah. And like where my mind starts to go is, yeah, if you use like heavy doses of broad spectrum antibiotics, it's not difficult to imagine you could just completely wipe out at least certain species of
hmm
bacteria from the gut of an animal. When you saw these diet induced shifts in the study we were just talking about, were you just were the populations shifting, or did you completely erase certain species with one diet or add brand new ones that weren't there before?
Yeah, that's typically how I think about it is that, at least when you think about macronutrient shifts in diet, that like it's basically like moving the knob on the
Yeah.
dial, and so yeah, it's rare that we see what, microbes that actually like drop below the limit of detection and are never seen again in the study. Um, we've looked at that kind of explicitly. We had a paper a while back where we had a really patient undergrad who switched the diet of mice from low fat to high fat every 3 days.
Oh wow.
Um, and so, it's relatively short intervention, but we were looking for cases of microbes that, showed a kind of hysteresis or memory of past diet. And we could find some examples, but they were rare. But that there is data from other labs like around Elenov, for example, has shown that like if you keep mice on a high fat diet long enough, um, that then when you return them to a low fat diet, some of the microbes look like they're gone. They're not able to recover.
I see.
And so
Yeah.
it might be kind of an effect of, how extreme, how strong is the dietary pressure, and then also like how long you
How
keep it
long does it
on before
last?
you.
Yeah.
But,
Interesting. Yeah. So you can get permanent changes with drastic interventions, whether that's
right.
using antibiotics, which are obviously an unnatural stimulus,
Mhm.
or these sort of artificially long dietary shifts.
Right.
I mean, my mind immediately goes to thinking like, well, obviously in the wild, animals in most places, including mice, as you mentioned before, they're seasonal,
Mhm.
right? There's four
Yeah.
seasons
Yeah.
in the year. Food is naturally going to shift. Maybe the body is sort of expecting or adapted in some evolutionary sense to shifts that happen on that seasonal time scale, but not ones that are lasting, longer than that.
Right. Yeah. Exactly. Yeah. And I think it's harder to kind of maintain these like long-term pressures.
Yeah.
And the other thing that I think is really, maybe this is good news for humans with the, um, we're also like we have constant exposure to microbes from other people,
Yeah.
and so, I think it's harder to kind of maintain permanent uh, disruption to the microbiome because we're constantly being reseeded.
Yeah.
Um, and that's been nicely studied in mice where if you treat a mouse with broad spectrum antibiotics and keep it alone in a cage versus in a cage with um, an untreated cage mate, the kind of co-housing allows you to, allows the microbiota to recover really quickly after a threat.
Yeah. So bottom line, based on all the stuff you mentioned so far, the microbiome, even when it's depleted, it can be regenerated, and it's very plastic and very responsive. It can shift very quickly in response to lifestyle changes such as dietary shifts.
hmm hmm Yeah, that's definitely kind of the working model.
Yeah.
um, and yeah, but I definitely think it like in terms of the some really simple things that we still have yet to follow up on from that study is, kind of like modeling better what the gradient would look like if you were on a less extreme version of of the animal-based diet.
Yeah.
Um,
Yeah.
or vice versa, if you extended it longer in the time, it'd be really interesting to see if these effects compound.
Yeah. Yeah. One of the things I wanted to ask you about that study is, so on the animal based diet, I believe you saw the microbiome shifts that you saw involved a lot of, you saw more bile tolerant organisms because presumably the animals were secreting more bile. Can you unpack some of the basic biology there when you're eating a ketogenic or truly high fat diet like that? What is that bile doing, and what is sort of the basic biology there?
Yeah, this is something, I guess I'm always like, excited when we see something in a human study that matches what we have seen in model organisms. And so Suzanne Devkota, who was in Eugene Cheng's lab at University of Chicago at the time, had this really beautiful paper where she, kind of getting back to your earlier question, they were interested in the question of whether or not microbes in the gut care about the type of fat.
Mhm.
Um, And so they fed mice high fat diets that varied in the source of the fat. And I probably will butcher it if I think it was the milk derived saturated fat if I remember correctly, but they found on kind of one of the high fat diets that there was this expansion in kind of a weird microbe named Balaphila wadsworthii. Um, and this, to microbiologists, like it's an interesting organism because actually named for loving bile.
Hmm.
It grows a lot better in the presence of bile acids, which is the opposite of most bacteria. Bile has a really strong detergent and antimicrobial effect. And so, it's thought to actually be one of the pressures that kind of keeps the level of microbes down in the small
I
intestine.
see. So bile is essentially acting as a detergent, and naturally, you would expect any cell-based organisms probably not going to tolerate too much bile because
Right.
everything's made out of a membrane. But
Uh-huh.
nonetheless, there are somehow, some way, nature has evolved certain bacteria that like the bile, basically.
Yeah, exactly. And specifically what Balafolo does, or one of the things it does to bile is that our bile acids in the gut have kind of decorations that are added by our body, including taurine. And so
Hmm,
Balafolo grows on the taurine. It
really?
can, cut it off of the bile acid and then use it as a substrate.
Hmm.
Um, and so, that was kind of just like interesting from a more micro basic side, but I think the reason why that paper got a lot of attention is that Suzanne went on to show that this expansion in Balafilla actually made mouse models of inflammatory bowel disease worse. Um, and
I see. So
so,
more having more of the bile tolerant species exacerbated this inflammatory bowel disease
right, yeah,
model in
exactly.
mice.
Yeah, and I haven't followed that literature carefully, so I don't know what the latest is in terms of whether or not that's still the case, and or how exactly that works. At the time, I think there was some speculation that it had to do with hydrogen sulfide.
Mhm, mhm.
Um, but yeah, we got interested in it just because when we looked at the animal-based diet, we saw one of the microbes that really went up was Bolophila.
Mhm.
And so that kind of made us wonder if maybe this elevated dietary fat intake could have kind of selected for this microbe that acts on fat
Hmm.
or that metabolizes bile acids. And the other thing we knew from the literature is that bile acids are controlled in that they're often produced in excess as we increase our fat intake.
I see. So
Um,
bile levels tend to go up. And correct me if I'm wrong. So the liver makes the bile, the gallbladder secretes and stores the bile, and the bile acts as a detergent. And basically, if you're eating a lot of fatty food, it's meant to sort of emulsify and act as a detergent
right.
for to handle all of that fat.
Yeah, exactly. So it's it's a good thing. We need the more fat we eat, we need the bile to kind of help with digestion. And so that we knew, and so that kind of like um prompted us to kind of look more carefully, and yeah, we were able to kind of consistent with the mouse study, um, show these increases in the level of the microbe that were associated with changes in bile acid composition.
Mhm, mhm. And you've done work. I want to talk about autoimmunity and inflammation, especially as it relates to the
Sure.
gut. You've done work in this area, and I'll let you kind of direct what work is most relevant to talk about from your lab. But before we dive into that, can you just give us a sense? So, but based on everyone I've talked to on the podcast and stuff I've read, basically inflammatory gut issues seem to be on the rise, and they have been, I don't know, as far as I can tell, for quite some time. To what extent is that true, and what are sort of the most common inflammatory gut issues that we see today in people?
Yeah, I'm definitely not a gastroenterologist, so I don't know if I can answer that well, but um, yeah, I mean, I think that you've probably seen the graphs of, increased chronic disease burden. They kind of like, coincide to the decrease in infectious diseases that are now, at least currently being controlled,
Mhm.
um, with antibiotics and vaccines. Um, but the um, Yeah, and so I think the kind of primary diseases that people are really focused on with respect to the gut microbiome are, inflammatory bowel disease, um, colon, as well as colon cancer, kind of linked to
Hmm.
inflammation,
Yeah,
and
yeah.
then, even diseases in other tissues, like we're working a lot now on multiple sclerosis, which is a disease of inflammation in the central nervous system that um, we think has links to microbes in the gut.
Hmm. Oh, really? Yeah, I mean,
Um,
inflammatory bowel disease, when you think about the gut microbes and food, I mean, inflammatory bowel disease, things like colitis, that makes sense, right? You're talking about the gut,
mhm,
the food goes through the gut, the inflammation's there,
right.
it makes sense. But you're saying you're working on multiple sclerosis. This is a central nervous system thing. And even that is potentially connected to gut microbiome stuff.
Yeah, so what's interesting is that I mean a lot of this has been studied with respect to how microbes in the gut affect the immune system, and so that is really well established within the gut. We know that if you look in a germ-free mouse, for example, they lack many of the kind of key immune cells that are typically found in the small and large intestine.
Hmm.
And then people have identified specific microbes that can um, kind of control the levels of different immune cell subtypes. Um, but the effects of the microbes in the gut it doesn't stay there because the immune cells can move.
I see. I see.
And then the other thing that happens is that microbes produce metabolites,
That enters
some of which
circulation.
we've talked about, like these short chain fatty acids, that can go into circulation and then reach other tissues. And so there's kind of this potential for microbes to remotely control immune function in many different tissues.
Yeah, yeah. So just
Um,
because the gut microbiome is, microbes in your gut, they can affect things anywhere else in the body potentially because the immune cells that might be in your gut at certain time points can literally move away. They are mobile,
mhm, mhm.
or the metabolites produced by the microbes can enter your bloodstream and then get all over the place.
Yeah, yeah, and then there's also, although we haven't really worked on this in our lab, but a lot of interest in microbes also moving throughout the body, and so the in certain situations like gut inflammation, you might there's some thinking that the microbes might actually translocate out of the gut, and
Oh
so
yeah. So like
you
if
may
there's
get microbes moving throughout the body.
I see.
Um,
So if there was, say, some inflammatory condition and say the barrier between the
right,
gut lumen and the inside of the body is compromised, then you start to get creatures that are supposed to be in the gut lumen and maybe they infiltrate and get in the body where they're not supposed to be.
right, yeah, exactly, yeah. Or another situation, if you have a lot of immune suppression, then the normal kind of mucosal defense that you have in the gut might not be able to keep the microbes out.
Yeah, yeah.
So yeah, so there's definitely like many different ways that we think microbes that are normally in the gut could affect other tissues.
Hmm.
Um,
I mean, some of the work you've done recently in the last year or so was looking at giving ketogenic diets and how that affected neuroinflammation and
mhm.
things like that. Can you walk us through some of that and also what the thinking was for why you went down that route?
Yeah, so we got really interested in that. Kind of starting on, to back up, we had done the study that we've been talking a lot about where we put mice on, or humans on an animal-based diet. And, we weren't really intending to study ketogenesis, but we did note that the subjects were in ketosis. But, it got me really interested in ketogenic diets, just because I guess I'm attracted to these more kind of extreme interventions. And we had shown previously that if you kind of take a mouse from a low fat to high fat, we did these gradient experiments and showed that it was a really predictable effect on the microbiota, kind of as you titrate up dietary fat. But that was all within the realm of kind of standard low fat to high fat. And so, whereas ketogenic diets are different in many ways. And so I wanted to more explicitly look at that and was, fortunate to get in contact with a group of obesity researchers that were designing a study that was really designed to look directly at ketogenic diets. And they were mainly focused on energy expenditure, but were kind enough to kind of collect stool samples from their subjects. And so we were able to look at subjects under really controlled conditions. These are all overweight and obese people given a ketogenic diet for 4 weeks, or kept on a control diet for 4 weeks
Mhm.
while they
Do,
were kind of living in a
yeah.
study center.
And let me just ask before you get into the results, do you know any of the details of the ketogenic diet, like the fatty acid profile or anything like that?
I do, yeah. I think some of that is in. I don't know if it's in our paper or it might be in the original study paper. But yeah, that was one of the great things about that study is that they at least in terms of energetics, they measured exactly what every single person ate for every meal.
Mm-hmm.
And then they did that not only in terms of like cataloging the food, but also measuring calories that remained. And so because the primary goal of the study was to kind of see whether or not energy, your kind of efficiency of energy extraction from the diet versus energy expenditure changed.
Mm-hmm.
But so we have a lot of like really detailed diet data from that study. We Yeah, we should probably return to that. I think we didn't. The analysis that we published was pretty straightforward. We kind of like left it at the two arms, but we didn't really do a lot of work like looking at the variation between people. I guess part of the challenge is that it was a relatively small number of people, so we we didn't we maybe need a bigger study if we're going to start getting into exactly which items people were eating.
Mm-hmm.
Um, but yeah, what was interesting is that we we're able to see clear differences in the human microbiome on this ketogenic diet relative to control. And then that kind of prompted us to start modeling this in mice. And so we could, although the results were different in many ways, one thing that kind of repeatedly kept coming out is that we saw lower levels of bifidobacterium, a group of gut bacteria, when subjects were on the ketogenic diet. Um, and so this kind of raised the question of whether or not bifidobacteria, is that a good thing or a bad thing?
Yeah.
Um, and at kind of a year or two before we were thinking about this, there was a nice study from Diane Mathis's lab at Harvard where they had screened, they did a very laborious screen where they took, different groups of germ-free mice and just colonized them one at a time with different gut bacteria, and then measured the levels of immune cells um, in the gut as well as in other tissues as a way to kind of figure out like which microbes affect which immune cells. And one thing they saw is that bifidobacterium in the gut can drive the expansion of T helper 17 cells, which we know are really important, both for fighting off infection as well as potentially playing a pathogenic role in autoimmune disease.
Mm-hmm. So
Um,
a type of white blood cell is responsive to the composition of the microbiome. When you go on a
mhm.
ketogenic diet, this one type of bacteria goes up. This is related to a type of white blood cell going up, and that could potentially be a good thing or a bad thing depending on if the conditions.
Yeah, actually, the opposite. So the ketogenic diet was driving the bifidos down,
Okay, down.
and and then we confirmed what Diane had shown, which is that at least some bifidobacteria, kind of drive increase the level of Th17 cells, and
I see.
so
And if that happens,
um,
the idea so if this ties to autoimmunity, so you don't want too many of these.
yeah, yeah. I mean, it's kind of a potentially a double-edged sword with
Yeah, yeah.
it raises questions about if you do have this happening while you're on a ketogenic diet, does that affect your risk of infection, at least gut infection, or does it potentially protect you from diseases like autoimmunity?
Yeah, yeah, and a
Um,
lot of I would imagine a lot of the stuff is sort of a Goldilocks thing, where especially when it comes to
right.
the immune system, you don't want it cranked up too high. You certainly don't want it too low. It's
Hmm.
autoimmunity on one side and infection on the other.
Right, exactly. Yeah, and we haven't really explored the infection angle, although that maybe is what we should have started with. With
Mhm.
in the initial paper, we really just didn't get to any disease model. We were just able to show the other thing that we found, which that was really interesting, is that you can drive the bifidobacterium down just by giving the ketone body. And
Ah,
so, you don't actually have to be on the diet. You can just the
I see.
elevation in the ketone body is enough.
Interesting. Yeah, I wanted to
Um,
ask you about that because that is a theme that I've seen come up across organ and model systems
mhm,
where, like, for just to give one example that will tie into this, completely different field, completely different organ. I was
mhm,
talking to a kidney biologist named Thomas Wimes, and he observed some very interesting things about how a ketogenic diet had protective effects for the kidneys in mice.
mhm.
And long story short, they show that BHB supplementation on its own, without going into ketosis, without having a ketogenic diet, just the BHB supplemented was able to completely recapitulate the protective effect on kidneys. And you're saying something similar is going on here, where the effect of the ketogenic diet on this white blood cell phenotype can be recapitulated by just giving them BHB.
Right. Yeah. Yeah. We looked at both the effect on immune cells, but also the changes in the gut microbiota.
I
And
see. So,
yeah, it's same experiment or similar experiment
mhm.
to what he did. We are using these ketone esters that are at the one we were using at the time was beta hydroxybutyrate with kind of attached to two medium chain triglycerides,
Hmm.
and that allows you to just spike it into the diet, or take it as a drink if you're a human.
Yeah, yeah, yeah.
And you can kind of artificially elevate ketone bodies without being on a ketogenic diet.
Hmm.
Um, and so,
Is
that's really exciting from the standpoint, both for the translational implications, but also, um, it just allows us to really simplify the experiment. And so,
right, right.
because now we can take out all the complications of changing fiber and, like all of the
Yeah,
other kind of things, baggage that comes along with a ketogenic diet in a mouse or human study.
yeah. It's pretty wild that just supplementing with that one molecule BHB can recapitulate at least most of what you see by this, what is otherwise a pretty drastic change going into a ketogenic state from a dietary intervention. It's a pretty, lots of things are changing when that happens.
Yeah. Yeah, I wouldn't say most necessary. I mean, I guess we still haven't like well
Yeah.
quantified that, but at least in terms
The
of
major things you focused on
the immune cell levels, it was very comparable. I think on the microbe side, definitely qualitatively, like
directionally
the
similar,
diet
but not
had a bigger
identical.
effect,
Yeah. Yeah.
So yeah, yeah, I wouldn't say we've like I think we are missing things,
Yeah.
and
Yeah.
it's an open question kind of what part of the ketogenic diet should be added to the supplement,
Yeah. Yeah. Okay. So,
But
so BHB wasn't fully reproducing the effects
right,
of the ketogenic diet. It was just,
yeah,
it was partially, but still pretty dramatically.
and really specifically, we saw this like suppression of bifidobacteria, which was very interesting.
Hmm.
And so, and we're still trying to kind of figure out why this happens. I mean, so we know that BHB has, all of these different signaling effects on the host. So, it could be that the, you know, compound is kind of directly changing the immune cell. But we all, we're a microbiome lab, and so we're more interested in, how the microbiome, what is it that kind of links BHB levels to changes in the microbiome?
Yeah.
Is that a direct effect? In the initial paper, we showed in vitro that you could change bacterial growth by adding BHB. And so, it could be that the compound itself has kind of an antimicrobial, or maybe even could benefit some microbes. Or alternatively, maybe the compound is kind of changing other things in the gut
Yeah.
that microbes care about.
Yeah.
Um, but yeah, that initial study or that paper kind of like set the stage for thinking about like which disease model this would actually matter in. And so um, we could have gone down the infectious disease route, but Maggie Alexander, who was a really talented postdoc working in the lab, um, was very interested in autoimmunity, and so we opted to go for this really well studied mouse model of neuroinflammation, where you kind of like prime the immune system with a
Mm-hmm.
combination of this peptide MOG and pertussis toxin,
Mm-hmm.
and
Mm-hmm.
that leads to kind of paralysis phenotypes that are similar to what patients with multiple sclerosis experience.
Interesting. Do you think, a little bit of a speculative question here? Obviously, that you just described these results where ketogenic diet or BHB supplementation can modulate the activity of the immune system. It has implications for neuroinflammation, autoimmunity stuff. In the realm of therapeutic ketogenic diets, in that field broadly speaking, there's a lot of excitement and some new studies around using the ketogenic diet for things like bipolar disorder, other neuropsychiatric conditions. Obviously, the ketogenic diet, I think its original claim to fame is that it's been known for a very, very long time that it treats certain forms of epilepsy. Do you think there's maybe a connection here that ketosis, a state like that produces high BHB levels, the BHB is just having very general effects in the body when it comes to inflammation and even nervous system function?
Yeah, it's a good question. That yeah, the epilepsy. So some of the really nice work on epilepsy that I can't really do justice, but it was actually a lot of it was performed by Christine Nelson, who's a postdoc in our lab right now,
Hmm.
when she was in Elaine Sau's lab down in LA. Um, but yeah, so there's also data kind of linking the microbiome to some of the epileptic benefits of ketogenic diets. Um, but yeah, I think it's still kind of early days. I think, even in mice, we don't really have a good sense for, how many situations, at least kind of limiting to the microbiome effects of the ketogenic diet. Like, is that always a good thing? Are there diseases where diet is changing the microbiome in a way that is actually not helpful? Um, and then um, yeah, I think that the mechanism is also difficult to unpack. Even though we've like narrowed it down to a single compound, part of the benefit as well as the challenge of studying BHB is that it has all these different targets in the body. And so, it's really hard to unpack that
Mm-hmm.
and to
Mm-hmm.
figure out, you know, how this is all working. But
Yeah.
but yeah, I mean, a lot to keep working on.
Has obviously there is no user manual here. We don't, even people like you in this field can't tell us, you should definitely do this or not do this to have a healthy gut microbiome. It's probably even challenging at this point to define precisely what is a healthy gut microbiome. What does that even mean for a given person, for a given context, for a given
Mhm.
state of metabolic health, all that stuff. Just broad strokes, as a microbiologist who studies this stuff, has this being in this field affected your relationship with food and antibiotics or anything like that in a particular way?
Um, Yeah, I mean, I guess like I'm the type of person where the more I learn about something, the less I feel like I know about it. So, not to use that as an excuse, but um, yeah, I mean, I think it's like it's hard to, given the state of knowledge, to like really convince myself that, I'm always second guessing, I guess, what I think the right diet is. So.
Mm-hmm.
I mean, it definitely makes me think a lot about what I eat, reading labels more than I used to. But yeah, I don't think it's really kind of dramatically reshaped what my own diet is.
Hmm.
Um, and yeah, I wish I could say the opposite. I hope that at some point we can get to the point where we can really translate this information to more actionable recommendations for
Hmm.
people. Um,
Given given what you just said, what do you think about this? So there's lots of products now that people can buy. I've had people on the podcast that work on these things. What do you think of these, microbiome products like Biome and like other things? Or, you could send in for a kit, you send them samples of this, that, or the other from your body, and then you get an analysis of what you're eating, and it tells you, you should take more of this, you should avoid eating that. Do those things really work? Is that information likely to at least be directionally accurate, or are we just not there yet?
Yeah, I wrote a commentary kind of, and I don't want to target any specific companies, but a couple years ago. Yeah, I guess my kind of opinion is that um, I think the thing that we're really good at right now is profiling. So, we can definitely kind of measure which microbes are in your gut. We can, look at their genes or metabolites. So we have a lot of tools in terms of characterizing your microbiome. I think, it's always the challenge of, what we do with that information. So that, even in kind of best case scenarios, the links we have between the microbiome and health are kind of at a more population or general level.
Mhm.
And so, like you have, I mean, it's the kind of classic challenge in medicine is like, it doesn't really matter what's good in general, it matters what's good for you. And
Yeah.
so, I definitely don't think we're at the point where we can accurately tell you what the best thing to do is. I do think it's exciting that, if at least some of these companies are often offering like repeated measurements of your microbiome. And so, I think there's some potential to, it kind of, if you think of it more as an experiment that, you start with a certain profile, maybe the company or your own, on your own initiative, you try to do certain interventions to improve it. And then you can kind of see what's working and what's not working. I think that kind of like paired diagnostic and intervention is really attractive, but it's definitely like I would consider it more in the R&D stage.
Mhm. What um what are some of the big questions or exciting things you're working on today in the lab that you're really excited about?
Yeah, so I think the topic that we haven't gotten to yet is really, how and is related to what you're asking about food. Um, is, how do differences in the microbiome kind of control drug response? And
Mm,
so, we got interested in this area, kind of similar to why we're excited about studying BHB as opposed to ketogenic diets. I think, when you think about pharmacology, it kind of narrows the scope to compounds that we know are really important for the body, and it kind of allows us you to then ask questions about how do differences in the microbiome affect either the level of those drugs or how they're metabolized and removed from the body,
mhm.
or
So
how they
the
make it to the target
yeah,
tissue.
the general idea here is if you take a pharmaceutical drug, say the gut microbiome could be important for activating that pro drug into the active drug. It could be important for how quickly the drug is metabolized and leaves your system, and all of that type of stuff.
Yeah, it's like, it's interesting to me, like really back to the very early days of drug discovery, there were links between the microbiome and drugs. And so, for example, one of the first antibiotics that was ever developed is called Prontosil. It has what's called an azo bond, which is two nitrogens with a double bond in between of them. And those azo bonds are not readily broken down by our own body, but bacteria can act on them. And so, and then it turned out that the active compound in Prontosil is actually the metabolite. And so you
Hmm.
need a microbe to actually create the antibiotic.
Hmm.
Even though, the drug that's given is effectively inert.
Yeah, yeah.
Um,
There could be, you could imagine scenarios where I don't know, maybe someone's doing a clinical trial, they're giving some drug to 100 people, and just we'll just make up numbers here. Maybe it works for 50 of the people and doesn't work for 50. It could be that what explains that is the gut microbiome and whether or not you've got the right bacteria to say turn the pro drug into the active drug.
Right, yeah, exactly. And also in the opposite direction. So, we have, I mentioned digoxin earlier. That's a classic example in the textbook that is a drug that is not really thought to be metabolized very much by the host, but is metabolized by bacteria to a downstream compound called dihydrodigoxin, and that metabolite is no longer active. And so you can have microbes in the gut that are both turning on and off drugs in ways that could change what amount of drug actually makes it to whatever the target is in the body.
Hmm.
Um, and yeah, so that's something we're really actively working to try to unpack for various drugs of interest.
Well, we've already covered quite a bit. Is there anything you want to reiterate or think we should cover off on from our discussion that you want to say anything more about?
No, I think that's great. We talked about a lot. So
All right. Well,
thanks for
Peter,
having me.
yeah, Peter Turnbaugh, you've done a lot of really good experiments. Some of the recent stuff I really liked. And also apologies for digging up that 2014 paper
No
from
problem.
your past. That was an interesting study, I thought. Thank you very much for your time.
Thanks, man.
I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.